Okay, so you've seen some of this information in class, but I decided to start at the beginning of this PowerPoint because it's been a long time since you've seen the information. So this, and the next few videos will cover the muscular system. And the big picture here is that any muscle tissue, the entire purpose is for it to contract and then return to its original shape. So during this video, we're going to learn about the gross and the cellular anatomy of muscle tissue. In later videos, you'll learn about the stimuli that trigger contraction. In another video, the molecules that carry out contraction. And in another video, the consequences of physiology to large-scale function. Okay, there are 4 tissue types. Muscle is one of them. And we will take a look at mostly skeletal muscle in this chapter. We will touch on the mechanism of cardiac muscle and the mechanism of smooth muscle. Okay, so there are 4 properties of muscle tissue that enable it to carry out its function. One is electrical excitability. This means that there is a current that is present across the membrane. So it's the membrane of muscle tissue that has a difference in charge between the inner and outer membrane. This electrical excitability is basically the reason why tasers work. Okay, contractility, or the ability to shorten, is a property that allows muscle tissue to carry out its function. Extensibility, in other words, the ability to stretch past the resting length without damage. And elasticity, which is the ability of the muscle to return to its original length after it's either been shortened or lengthened. So these are the 4 properties that allow muscle to carry out its function. Okay, so we're going to take a look at the anatomy of skeletal muscle tissue from the muscle as a whole all the way down to the protein components of skeletal muscle tissue. So the first thing to point out is the periosteum, which is actually part of a bone. This is a connective tissue sheath on a bone, and it's very tough. And the important thing here is that the periosteum is continuous with the tendon. One thing I want to point out here is that I'll have the connective tissue on this slide labeled in blue. So the tendon is connective tissue. Okay. Next we have the epimysium. If you look at the at where the line is pointing, the epimysium is a connective tissue sheath that surrounds the entire muscle. So epi meaning outside and mysium referring to muscle. This next label is the belly of the skeletal muscle. So this is the point at which the skeletal muscle is at its widest. Next up we have a fascicle. So there are multiple fascicles in each skeletal muscle. Each fascicle is surrounded by perimysium, which is a sheath of connective tissue surrounding, again, the fascicle. Within each fascicle, you have clusters of muscle fibers or cells. Muscle fiber and muscle cell are interchangeable terms. Within the muscle fibers, surrounding the muscle fiber, you have another connective tissue sheath, which is not pictured here, called endomysium. We'll see that in later slides. And within the muscle fiber, you have myofibrils, which are the organelle of contraction. More detail about that later. So one thing I also wanted to point out, that all 3 layers of connective tissue, the epimysium, the perimysium, the endomysium, can combine to form tendons. And tendons are what connect muscle to bone, as opposed to ligaments, which connect bone to bone. Now, if you ever find yourself mixing up tendons and ligaments, just remember your most well-known tendon, the Achilles tendon, which attaches your calcaneus bone to the gastrocnemius or calf muscle. Sometimes the connective tissue forms a much broader sheet, a broader, thinner sheet. It's no longer rope-like. It is definitely sheet-like. This is called an aponeurosis, and aponeuroses usually connect muscle to muscle. Next, we're going to take a look at the organization of a fascicle. So first you have the perimysium, which is that, again, tough connective tissue sheath around the fascicle. Fascicles are bundles of muscle fibers, and again, remember that a muscle fiber is a muscle cell. One other thing I want you to remember is that muscle fibers run the entire length of the muscle. So from tip to tip with the muscle fibers run that entire length. The individual muscle fibers are surrounded by connective tissue called endomysium. The sarcolemma is a membrane that surrounds each individual muscle cell or muscle fiber. Within the muscle fiber you have cytoplasm, which is called the sarcoplasm. Within the sarcoplasm you'll find mitochondria, which are responsible for production of ATP, myofibrils, which are the organelle of contraction, and again more detail on this later, and nuclei. You have multiple nuclei with each skeletal muscle cell. We'll see the reasons why in just a minute, but I want you to think about this. Nuclei produce mRNA, which is then transcribed into, nuclei are where DNA is transcribed into mRNA. The mRNA is then translated in this case, the sarcoplasm, into proteins. Now, given that skeletal muscle fibers run the length of the muscle and that myofibrils also each run the length of the muscle, there's a real need for a lot of protein production. And so multiple nuclei are going to be very handy for this. Finally, we have another cell called a satellite cell. More detail on this function later. Now we're going to take a look at a muscle fiber. So first up we have the sarcolemma, which is the outer membrane. Sometimes the sarcolemma is continuous with the endomysium. Next we have the sarcoplasmic reticulum. This is a specialized smooth endoplasmic reticulum. Now in most cells, the endoplasmic reticulum, the smooth ER, is responsible for detoxification in the cell and production of lipids. In this case, the sarcoplasmic reticulum serves as a reservoir or storage spot for calcium. Next up we have a triad. Now a triad is a combination of a couple of different things. First of all, terminal cisterns, which are the points of the sarcoplasmic reticulum that come into contact with T-tubules. Now the T-tubule or transverse tubule, I want you to look at the color of this. It's the same color as the sarcolemma, and actually the T-tubules are where the sarcolemma penetrates down into the cell. The reason for this will become very evident later on. Here are mitochondria again, and skeletal muscle cells are actually pretty packed with mitochondria. More mitochondria in active individuals than in individuals who are sedentary. Next we have the nucleus, and again, multiple nuclei in a skeletal muscle cell. We have the sarcoplasm, which is the cytoplasm that contains all of these organelles we've been talking about. This bracket shows you the myofibril, and we'll go into detail about the myofibril right now. Okay, the myofibril is composed of thick and thin filaments. They're made up of different proteins, which we'll talk about in just a minute. The sarcomere is the contractile unit of the myofibril. So basically, in the sarcomere, these structures, the Z discs, are pulled together, and as each sarcomere within the myofibril shortens, the entire myofibril itself shortens. And when the myofibrils shorten, the muscle fibers shorten. When the muscle fibers shorten, the muscle itself shortens. Okay, so we're going to run into a little bit of repetition here, but I don't think there's much wrong with that. So because muscle cells can be pretty long, their average length is 4 inches. They're called muscle fibers. And so again, for this entire chapter, fiber and cell are going to be interchangeable. So myofibrils, as I said, run the length of the muscle fibers. They contain myofilaments, thick and thin, which run against each other. And it's actually the thick myofilaments that pull on the thin myofilaments, which brings the Z discs together, shortening the sarcomere. Okay, so again, these are terms that you've seen. The myofilaments, thick and thin, they move against each other, they contract the sarcomeres, and then therefore the muscle fibers. Okay, now I said that we would discuss the role of satellite cells. So basically, satellite cells are going to be involved in repair of skeletal muscle tissue, and we'll get into that in a little bit more detail in one of the later videos. Okay, but the other thing that I wanted you to see, or the main thing that I wanted you to see with this particular slide, is that myoblasts actually fuse into a skeletal muscle fiber. So the reason why skeletal muscle cells have got multiple nuclei is because they are themselves made up of multiple cells. So each cell had its own nucleus, contributed a nucleus to the skeletal muscle fiber, so that ends up with lots of nuclei in one very large cell. Okay, so this is another picture as a reminder again of the cellular anatomy. So what we just took a look at was a quick look at the biofilament. Okay, now this is what's called the microscopic anatomy of the muscle, and so I want to point some things out here. Look for the blue arrows. So right here, this is the sarcoplasmic reticulum. This again is a storage reservoir for calcium. It's got a very, very high concentration of calcium in it, about 10,000 times the calcium concentration found in the rest of the cell. So this is actually an active transport pump for calcium. And so in order to get as large a concentration of calcium as you have in the sarcoplasmic reticulum, you're going to have an active transport pump to work to push calcium against its concentration gradient into the sarcoplasmic reticulum. These little arrows represent calcium release channels. Most of the time these release channels are closed. When they're opened, and we'll talk about the stimulus that opens them in the next video. When a stimulus opens them, because the calcium is at such a high concentration within the sarcoplasmic reticulum, the calcium rushes out really fast. One thing that we talked about in lab when we talked about diffusion was we said that the steepness of the concentration gradient determines how fast that diffusion occurs. And with a 10,000 to 1 ratio, diffusion happens very fast. And just the millisecond that this calcium is released from the sarcoplasmic reticulum, it will flood the entire cell immediately. You'll see the importance of that later on in this chapter. This arrow is basically showing you a myofibril, and again, this is the contractile organelle. This arrow is showing you a sarcomere. Now I want to point out once again that there are multiple sarcomeres along the length of a myofibril. Think of these as sort of links in a chain. If you wanted to shorten a chain, if you shortened every link in the chain individually, then the entire chain itself would shorten, and that's basically the role of sarcomeres. This arrow is pointing to a mitochondrion, and again, you have to have a lot of mitochondria because this cell uses a lot of ATP. So one way that it uses ATP is when those calcium active transport pumps get started up. So for every ion of calcium that gets pumped into the sarcoplasmic reticulum, and there's a lot of them, you consume an ATP. Myoglobin is a protein that binds to oxygen. It's related to the protein hemoglobin, which carries oxygen in the bloodstream. Myoglobin serves as a temporary storage site for oxygen in skeletal muscle, so that if the oxygen supply gets low, then the muscle tissue can start taking oxygen from the myoglobin itself. This arrow is pointing to glycogen granules. Now you've seen glycogen before. It is a polysaccharide that is made up of glucose subunits. So one thing to remember is that cellular respiration requires both oxygen, which is supplied by the myoglobin, or can be, and glucose, which can be supplied by the glycogen granules in the skeletal muscle. So basically the idea here is if the cell begins to run short on oxygen or begins to run short on glucose, then myoglobin for oxygen and glycogen granules for glucose can serve as an internal storage for both of these very important ingredients of cellular respiration. Okay, so next we're going to take a look at the arrangement of a sarcomere. So the first thing to talk about is Z discs. These are at the very boundaries of the sarcomeres. So what I want to say here is this top diagram is a representation of myofibrils. It's a common representation of myofibrils. This bottom diagram shows you the thick and thin filaments in a little bit more detail. But regardless of which diagram you're looking at, the Z discs are the boundaries of the sarcomeres. And so you can see there's an attachment between the thin filaments and the Z discs. So when the thick filaments pull the thin filaments toward the center of the cell, then the thin filaments are going to tug the Z discs in towards the center of the cell. Okay, next structure is the A band, and if you look at the A band, it's pretty broad. So the A band contains both thick and thin filaments. So the entirety of the thick filaments and portions of the thin filaments that have overlap. Now, one thing that you've seen already this semester with skeletal muscle tissue is that it had striations, light and dark sort of stripes on the muscle. So the A bands are the darker stripes because with thick filaments and portions of thin filaments in them, there's more protein there. It blocks light better, so they appear darker on a microscope. Next structure is the I band. And the I band is the area, is a lighter area that contains just thin filaments. So with just thin filaments, there's more space in there. Light can come through it, so they look lighter under a microscope. Okay, next up is the H zone. And the H zone is the portion where you just find thick filaments. So if you're wondering, is the H zone part of the A band? Yes, it is. Okay, next up we have the M line, which is the region in the center of the sarcomere. Now, this region actually holds the thick filaments in place so that they have an anchor for when they're pulling on the thin filaments. The M line is also the very center of the sarcomere, and it's what the Z discs get pulled towards. This picture just shows an electron micrograph, transmitting electron micrograph of an actual muscle fiber. So here you can see some of the parts that we've just been talking about. You can see the M line in the very center. You can see the H zone, which is just the thick filaments. That much darker area is where there's, outside the H zone is where you have overlap between thick and thin filaments. So you see that the H zone is part of the A band. Bordering the A band, you've got the I band on either side of it, and this is going to be just the thin filaments. And you can see how much lighter that the thin filaments are even under an electron microscope. The Z discs are the boundaries of the sarcomeres. So one thing that I want to make clear here is that the A band is contained within one sarcomere. The I band is actually made up of portions of two sarcomeres. So look at the label at the bottom where it says sarcomere, and it's the edges of the bracket are pointing up towards the Z discs. So the remainder of the I band outside of the bracket where it says sarcomere, this is the beginning of two other sarcomeres. Okay, next we're going to take a look at the muscle proteins themselves. So there are a number of different functions with the muscle proteins. There's contractile, there's regulatory, and there's structural. Now when we go through the mechanism of contraction, you'll know what the roles of myosin and actin are to a much greater degree. So basically the myosin is going to attach to and tug on the actin. So if you look at the structure of actin in the middle diagram, they're the little yellow balls. And so these are actually attached together in a thin filament, and they actually look quite a bit like the microfilaments that we saw as part of the cytoskeleton, with the exception that they have two more proteins associated with them, and those are troponin and tropomyosin. So tropomyosin is that sort of brown colored fiber that you see. And what the tropomyosin does is to cover up binding sites on the actin. If you look at the actin molecules closely here, and you might want to pause the video and sort of zoom in on this, you'll see little black dots on the yellow balls, and these represent binding sites that can attach to myosin. Now, if the muscle is in its resting state, the tropomyosin actually covers up the binding sites. And what this does is to not allow any binding between thick and thin filaments. And if there's no binding between thick and thin filaments, the muscle cannot contract. So in this case, the tropomyosin is actually a regulatory protein that keeps the actin and the myosin from actually binding. Troponin is what attaches the tropomyosin to the thin filament and keeps it in a specific place. Now, if the structure of troponin changes, then the location of tropomyosin is going to shift. That's an idea that I want you to keep in mind for a later video. As far as structural fibers go, titin is a structural fiber. Okay, and if you look at the bottom diagram, the titin is that green sort of spring-like looking protein. Now, the idea that I want you to have in mind here is basically like car springs. So when they're compressed, they'll spring back into place because they're elastic. So titin is an elastic protein. And when the Z discs are pulled in towards the M line, how do you return the sarcomere to its original length? Well, the titin actually springs back out and returns to its original shape. And so in this case, it's going to be called a structural protein.